So thanks for a nice invitation. And so I'm going to present the Brain Scaffold Builder, which is a framework that we have developed here in Pavia, and we have basically applied for the cerebellum, but then other groups and collaborators are using for other brain regions like hippocampus, some thalamic nuclei, and so in order to have a flexible framework, really easy to be also expanded with user-defined interfaces, let's say. And so this is a tool that we are using a lot. So this is the main structure. So we have 5 main components. So the volume, the cell types, the placement, connectivity, and simulation. This is a Python package, really easy to be installed. And here there are the links on GitHub and documentation. It will be soon a component in the EBRAINS. And it will be one also of the main tool for the cerebellar modeling hub in the University of Pavia. So this has 3 main interface for simulation with neuron nest and arbor. I will tell you a little bit later. So let's start with the network reconstruction. So first of all, we define the volumes. The volume here. So it is in the configuration file, so a JSON file. We can really define any sites, but even just using complex shapes extracted by brain atlases. In this case, in the cerebellar use case I'm showing now, we have used these sites. So we have defined specific sites on the axis, so sagittal plane, horizontal plane, coronal plane, of course constrained on the data. So we have specific thickness of the layers composing the cerebellar cortex with specific proportion, of course. So we keep these sites, for instance, thickness of the granular layer, 130 micrometer, then Purkinje layer in between, and then the molecular layer, more or less 150 micrometer. So then we defined how many and which cell types we want to put into this volume. So in this case, we have granule cell, Golgi cell, Purkinje cell, stellate cell, and basket cell, and then the glomeruli, which are basically the terminal of the mossy fiber coming from outside the cerebellum into the input stage of the network. For each cell type, it's easy to define one placement strategy, which is not necessarily the same for all cell types. And then we can define density values, planar density, volumetrical density, or even a count ratio with other cell types. And then a radius of the soma in order to avoid overlap. So this is the case of Purkinje cell, for instance. So we define more attributes. So we have a planar density because they have a specific placement fashion. And then we have some extension linked to the morphology because there are constraints on the distance which are not isotropic. So we need to define specific distances among adjacent cells which are related to the direction, the axis. Of course, each one can be associated to one or more morphology files, or we can also deal with something without morphology. And then in that case, of course, we need to apply specific connection rules. I will show you soon. So the third component is the placement. So for each connection type, we define the placement. We can also use voxel-based density values from atlases. And then in this case, for instance, we use particle placement algorithm for all cell types except the Purkinje cell. So we have a random placement, then check for collision, and then a sort of repulsion force if collisions are detected, and then pruning if some cells are outside the volume. While for the Purkinje cell, we use the planar array placement strategy with specific orientation and, of course, some stochastic noise, let's say, on the 3D position. And then we place this number in the volume I have defined, but then, of course, We have also used a shape from the Allen. So in this case, we have the floccular regions of the cerebellum, of the mouse cerebellum, the two sides, right and left, and then we have filled again with these cell types and specific placement strategy. So now in this use case, we have about 30,000 neurons with specific density in the different layers and even some anisotropic rule. Then we define the connectivity. So again, we have a lot of available strategies depending if we have or not detailed morphology, or we have, for instance, in some cases, we have few morphologies available. So we prefer to avoid, let's say, to overfit the synapse location with touch detection, but we prefer to use some strategies like voxel intersection in order to introduce a little bit more variability, since maybe we have few morphologies that they are a little bit too much repeated into the volume. So here, for instance, it's a case between granule cell in red with ascending axon and parallel fiber, the two parts of the axons. And we use touch detection to identify synapses on Golgi cells. So blue and light blue are two Golgi cells. And another case, we use voxel intersection between a stellate cell, which is an inhibitory neuron, the yellow one, inhibiting a Purkinje cell, a green one. So we voxelize the axon of the presynaptic neuron and the dendritic tree of the postsynaptic neuron, and we identify the overlapped voxels. Inside this voxel, we identify possible branches of the morphology on which a certain number of synapses can be placed. Of course, we can also define a sort of distribution of a number of synapses, so multiple contacts for each connected pair, in order to identify a certain plausible number of contacts. In some cases, we don't have any morphology, so we can use probabilistic rule based on proximity or on divergence convergence ratio, of course, using the distances. And we can, for instance, define group of cells excited by the same glomerulus with specific constraint. So we came out with a cerebellar connectome with more or less 1.5 million of synapses and also gap junctions. So the idea is that we unify a collection of scattered experimental data into a coherent construct, and so also some capability to predict in a model-based fashion some information about circuit organization. Then I go very fast. We have, of course, the simulation step, so we can interface with the same reconstructed network. We can go in neuron simulators, in nest simulator, or in Arbor, depending, of course, on the neuron models and synapse model that we introduce in the configuration file. In this case, we use the neuron simulator.
Is it muted or is it just
no? I think we she's back.
Yeah, we lost you for a moment there.
Oh, sorry. I hope not too much.
Just 20 seconds or so.
Yeah.
Okay. Yeah, it told me that my connection is unstable. Okay, by the way, I was showing some functional validation of the model with background frequency, synchronous oscillation in the granular layer, then specific pattern of responses like burst and pause in the Purkinje cell, the green one, and then specific pattern of inhibition from the molecular layer interneuron basket and stellate cells, orange and yellow, that create specific pattern, contrast enhancement, and other properties into the Purkinje layer, which are the main output, because then they project to the DCN to go outside the cerebellum. So then we can easily modify some properties, plasticity parameters. It's easy to be changed in the configuration file, simulating a sort of reinforcement, LTP, LTD. And then we found some dynamics propagation of the signals depending on specific realistic inputs, which is background in vivo and even whisker RPath in this case with specific stimulus on the mossy fibers, which represents a sensory-like stimulation. Then we can record, we have a Blender module interfacing with BSB to plot some cells into the network with specific synaptic current, ionic concentration along the simulation. So this is a Golgi cell and a granule cell with very simplified morphology for dendrites, ascending axon and parallel fiber. And so then we simulated similar input in Nest, so plotting raster plot and PSTH using simplified neurons and alpha-based conductance synapses. So, okay, thanks a lot. Sorry for running out the time. And of course, it's big work from a lot of people, experimentalists or computational modeling lab and other collaborators.